All right, well, thanks so much for having me here. I'm really excited to tell you about this methods paper, Testing for Underpowered Literatures. So key to reproducible and credible research is good research design. And a key question that anyone who is either designing or funding an experiment is going to have to confront is how large should the sample size of the experiment be? On the one hand, a larger experiment with a larger sample size will have greater statistical power, and it will be more likely to detect a treatment effect if one is there. But on the other hand, larger experiments can be more costly. This means that a funder of experiments would be justified in asking the following question: are we funding too many small experiments? Or to reframe this in terms of a counterfactual, the funder might instead ask, how many more treatment effects would we have detected if we had instead funded larger experiments? What I'm going to do today is to propose a new meta-analytic statistical method for studying this counterfactual and for estimating how many more effects a set of experiments would have detected if they had counterfactually been larger than they really were. And just to very quickly give you the flavor of where we're going and what we're trying to accomplish today, I'm going to mention one of my empirical applications right away. I applied the method to a couple of well-known meta-analytic data sets, and I find, among other things, that if we were to take the set of all experiments recently published in top economics journals, and we were to go back in time and counterfactually rerun all of these experiments, only with double the sample size that they were actually run with, then we would expect to see an increase in the fraction of statistically significant t-scores of about 7.2 percentage points. So you might wonder, is this a large increase? Is this a small increase? How should we think about that? And I'm going to present several benchmarks to you and argue that this 7.2 percentage point gain is a relatively small gain. But before we get to applications, let's take a moment and step back and think a little bit more carefully about what the research question is that we're trying to answer. So I want you to consider any population of experiments. This could be all RCTs published in top economics journals, or maybe it's all laboratory replications that were funded under a certain initiative. This set of experiments can be anything you want. They can be studying different interventions taking place in different places. They can use different methods of measurement. All that the experiments need to have in common is that they report t-scores. They don't need to be united by any other common theme. So our research question is going to be, how many more of these t-scores would have been statistically significant in expectation if we had gone back in time and counterfactually rerun all of the experiments only with X times the sample size? Another way of saying this is we're asking how much would average statistical power over this population of experiments have increased if we'd multiplied all of the sample size by a chosen X. Now, this is a very challenging research question to answer. It's really hard. And the reason it's really hard is this: how sample size maps into statistical power depends on the magnitude of the true intervention treatment effect. But the magnitude of the true intervention treatment effect is never directly observed. The researcher doesn't directly observe it. The meta-analyst doesn't directly observe it. To clarify why this is such a challenge, I'm going to present you with a simple simulated example. So consider two populations of experiments, two literatures. In literature one, all of the interventions that are being studied are identical. They all have the same true intervention treatment effect, and they're all moderately effective. Contrast this with literature two. In literature two, half of the experiments are studying interventions that don't do anything at all. the true treatment effect is very close to zero. The other half of the experiments in literature 2 are studying interventions that are highly effective with very, very large true intervention treatment effects. For these two literatures, sample size choice is going to map into average statistical power very differently. In other words, average statistical power is going to respond really differently to sample size choices in these two literatures. And the figure illustrates this contrast. On the x-axis, we have the sample size of every experiment in both literatures. And just for the purposes of this figure only, I'm going to assume that sample sizes are the same across every experiment, but we'll immediately throw out that assumption once we move on from this slide. On the y-axis, we have the average statistical power within each literature at that sample size. So for literature 1, which is the solid line, you can see that average statistical power responds very much to your choice of sample size. As we increase the sample size, average power rises rapidly. However, in contrast, for literature 2, which is the dashed line, as we increase sample size, average statistical power hardly responds at all. How is this even possible? Well, for literature 2, half of the experiments are studying interventions that don't do very much. The true treatment effect is close to zero. So increasing the sample size doesn't make you more likely to find something if there is nothing there to find. And in addition, for the other half of the experiments in literature 2, the interventions are highly effective, so effective that power is already 100% by the time we even started the figure. Power has nowhere to go as we increase the sample sizes. So that's why literature 2 is less responsive. What I'm trying to do here today can be boiled down to the following question. Can we determine from a sample of t-scores whether they were drawn from a population of experiments that responds more like literature 1 or that responds more like literature 2? This is a useful number to know if you're funding experiments, because then you would learn the upside of increasing your sample size requirements. So how is this possible? How can we develop a method in order to do this? Well, unfortunately, I don't have time to inflict all of the mathematics on you. So instead, I'm going to informally sketch out the way that the method works. And you can see the full mathematical proofs are available in the working paper online and in the transparency package online. But let me just illustrate to you the intuition for how this works. And we can think of this as happening in 4 steps. In step 1, we need to collect a sample. So we're going to collect a sample of n t-scores published in m articles. The t-scores are allowed to be correlated within an article, but they have to be independent across articles. So here we've collected an imaginary sample of t-scores in a histogram, and already you can see there's something wrong with this histogram. You'll notice there's a big jump at the critical value of 1.96. Something's up. It looks like we're missing some of our statistically insignificant t-scores. So in step 2, we have to remove the effects of publication bias from the histogram of t-scores. We estimate how large the discontinuity is at the critical value, and then smooth out the histogram in order to eliminate the effects of publication bias. So now we have a nice depublication biased histogram of t-scores. The third step is the challenging one. And this is where I'm glossing over a lot of the more complicated math. We're going to do a fitting exercise. What we're going to do is we're going to find the population distribution of true intervention treatment effects that best explains the histogram of t-scores. So here, the blue line is the distribution of true effects that best rationalizes our histogram of t-scores. And in the fourth step, we're going to compute the quantity that we really want to compute. We have an estimate of the population distribution of true effectiveness of the interventions in our population of experiments, but that's not what we really want. What we really want is the distribution of t-scores under a counterfactual, under the counterfactual that all the sample sizes are larger. So here, the red, and fortunately, it's a very easy conversion that we can make. If we know the blue line, if we know the distribution of true effects, we just integrate it, and there we get the distribution of t-scores under the counterfactual. So in this example, the red line is what the histogram of t-scores would have looked like had we doubled every sample size of all of the experiments in this population of experiments. So that's a sketch of how the method works. Let me just briefly point out some of the ways in which this method is contributing to the recent conversation on how to do meta-analyses involving t-scores. There's been a lot of really great recent work that, from one perspective or another, studies average statistical power over a population of experiments. But one feature that all of these methods have in common is that they make some sort of functional form assumption about the population distribution of true intervention treatment effects. For example, it's common to assume that it's possible to take our population of experiments and subdivide it into subpopulations, and within each subpopulation, all of the interventions are assumed to be identical. Another common assumption is that the distribution of true intervention treatment effects has a parametric shape, like a gamma distribution. And one contribution that's being made here is that I'm trying to show that it's possible to do without these assumptions and to go without any functional form assumptions at all about the population distribution of true intervention treatment effects. There's, of course, a price to be paid here. It makes our standard errors go up. But what's being demonstrated here is that it is possible to move forward without those assumptions. If you're interested in the math, this problem belongs to a class of problems called deconvolutions. This class of problems is very well known across the sciences. There's lots of methods for studying deconvolutions, but in this setting, the setting that we're all interested in, there's something going on, publication bias, and publication bias means that not every t-score is observed, and this makes it impossible to use standard deconvolution methods. We need a deconvolution method for this field. So those are some of the methodological contributions that are being made. So now that we've talked about the method, let's take it out for a drive and try to use it to learn a little bit about the real world. So in my first empirical application, I'm going to use the methods matter data set by Brodeur, Cook, and Hayes, and we're going to use it to study randomized control trials published in top economics journals. So this data set contains t-scores from more than 200 articles published in 25 top econ journals over a certain period of time. And all of these t-tests study main hypotheses of interest, and those hypotheses were all identified using either a randomized control trial or a natural experiment. They're all causal hypotheses. And so let me show you the results. So here I've plotted out the results just for RCTs in a graph. And let me tell you how to read the graph. On the x-axis, we have the amount by which we are counterfactually increasing all the sample sizes. So on the left at 1, we're not increasing any of the sample sizes at all. So we expect a 0 power gain. And on the right, all the way over on the right, we're doubling all of the sample sizes in this set of experiments. On the y-axis, we have the amount by which we've estimated average statistical power to have increased as a result of increasing all of these sample sizes. What we find here is that if you look all the way over on the right of the graph, you'll see that doubling the sample size of every experiment in this data set would lead to an increase in statistical power of about 7.2 percentage points on average. So that was the easy part. Now this is the hard part. Is this a small gain or a big gain? Should we think of RCTs published in top economics journals as a set of experiments that would really benefit a lot from increasing their sample sizes across the board? Or do we think this is a lackluster gain? And I'm going to argue to you with several benchmarks that this is a lackluster gain. So the first benchmark I'm going to use is to just look at the natural experiments in the same data set. So if you look at the natural experiments published in the same journals at the same time, you see that they are more than twice as sensitive to counterfactual sample size increases, more than twice as sensitive as the RCTs are. Now, and this difference is highly statistically significant. So this might not be so much of a surprise, though. for an RCT, the researcher does power calculations usually and decides on their sample size on the basis of those power calculations, we hope. Whereas for a natural experiment, it might not necessarily be possible to change the sample size of the natural experiment on the basis of power calculations because the experiment was naturally occurring. So it would make sense that RCTs tend to have better optimized sample sizes, at least in terms of power, better optimized sample sizes than natural experiments. But if that benchmark doesn't surprise you, then maybe this next benchmark might. So I'm going to apply the method to a second well-known data set. This is the Many-Labs 1 systematic replication study. And if you're not familiar, this data set contains data from 36 independent psychology laboratories, each of which were attempting to replicate the same set of published effects that had been previously published and found. And these experiments were not small. The smallest was 79 participants in the lab, and on average, they were more than twice as large as that. So there are two features of why are we using this data set? Why is this important for our purposes? Well, there are two features of this data that are very, very useful for constructing the benchmark that we want to construct. The first is that these are replication studies. They were designed on the basis of a previous experiment that the experimenter hopes is identical to the one that they're trying to run. And this means that we would expect these to be fairly well powered. Now, we might see some evidence later that replications aren't as well powered as we might like, but I would argue that it's still a good benchmark because for an RCT, usually the intervention is novel and you don't have previous data to design an experiment with. But for these replications, they were designed on the basis of a previous experiment, so we would expect them to be fairly well powered. The second feature of this data set that's really special is that the experimental interventions are meant to be identical across laboratories. That's the attempt anyway, to make experimental interventions that are the same from laboratory to laboratory. And indeed, the authors find that the variation across laboratories is very small compared to the variation across experimental treatments. So if we exploit that fact, that the interventions are nearly identical across laboratories, well, then we can vastly increase the precision of our estimates of the power gain. So now here are the results. And the blue line is the same as before. That's RCTs published in top economics journals, exactly the same as before. Now the red line is the Many-Labs replication studies. And you'll see that it's quite precisely estimated because of the features of the data we're exploiting. And these two power gains track each other quite closely. Indeed, they're statistically indistinguishable from one another. So we see very similar power gains between the RCTs, most of which are in the field in economics, and the Many-Labs replication studies. Okay, so how do we make sense out of all of this? How do we interpret this? What's our call to action here? Well, we've estimated that RCTs published in top economics journals would gain about 7.2 percentage points of power on average if we were to counterfactually double their sample sizes. This is off of a base of about 38% average statistical power without changing the sample sizes. And the standard error here is about 2.5 percentage points. And we're going to use three benchmarks to argue that this is a small power gain. The first benchmark is that quasi-experiments we find to be more than twice as sensitive as RCTs. The second benchmark is that the Many-Labs replications that we expect to be well-powered are statistically indistinguishable from the economics RCTs in terms of their power gain. Now let's construct a third benchmark, and we'll do this one with a thought experiment. Imagine that you've just designed an experiment, and luckily for you, your power calculations happen to be exactly dead on. Your experiment is exactly 80% powered to detect the true treatment effect. Of course, you can never know this, but let's assume that this is the case. If you were to take that experiment and double its sample size on top of what it already had, then you would expect to see an increase in statistical power of 17.8 percentage points. So even though this experiment was well-powered, it's still a lot more sensitive to counterfactual sample size increases than the average that we've estimated for RCTs in economics. So the takeaway here is if you believe these benchmarks, and there's of course other arguments that can be made in the opposite direction, but if you believe these benchmarks and if you were to accept this comparison, then you would conclude that RCTs in economics are fairly insensitive to sample size increases, at least in terms of their average statistical power. And what would the call to action there be? Well, if we had a bunch of extra money, we should probably spend that money on running more RCTs rather than increasing the sample sizes of the ones that we were already running, at least across the board. So the next step for this project is really so we've had some simple, these are some relatively simple examples looking at averages across very big populations of experiments, but we want more than big averages. Maybe there are subpopulations that are more interesting. And in particular, we want to work with funders to try to wield the method in a way that produces estimates that are useful to funders. So now we have time for questions. Thanks very much.
Thanks, Stefan. I have two kind of interrelated questions. That was really cool. The first is if really we did have twice the sample size on existing studies, then would it be the case that the distribution of observed treatment effects would change a lot? Like maybe with the sample size we have, a lot of treatment effects are disappearing because of publication bias. And maybe that's driving some of the results. Like you said, the distribution of those treatment effects tells us how steep that curve will be. So I don't know if you can speculate on that. I was trying to think through what it would look like if certain effect, like if lots of zeros are missing, maybe that's okay for you, because if you have a lot of zeros and a lot of large effects, that just flattens out the sensitivity curve. So maybe it's okay, but I wanted you to comment on that point. And second, I thought I really like the conclusion in the sense that I've had a lot of discussions with funders through the years who've said, we should fund half as many studies, but double the sample size because we have too many underpowered studies, and I never knew what the answer was to that. So you're giving us some guidance. Although again, it feels so sensitive to what this distribution of treatment effects is. So anyway, just congrats on the paper. And I'm curious about your thoughts about those missing estimates that are out there.
Yeah, so the estimate that I presented here, this is after the 7.2 percentage point gain is pre-publication bias. So if you look at the distribution of t-scores that are actually drawn, the real ones, before we've dropped the insignificant ones, the gain is 7.2 percentage points. The gain would probably be a little bit different if we then apply the publication bias. And that I don't have an estimate up there for you, but this is the true, if we killed all the publication bias, then that's the amount that we would gain. Now you asked about the distribution. Yeah.
I thought your distribution of t-stats was from published studies. So I'm saying that so how are you getting the true effects when there's all these unpublished studies that we never observed in a Bell's data set? Are you making assumptions to fill in the distribution then?
Yeah, so the assumption is that publication bias follows a certain form. So the methodological assumption here, which can be loosened a bit, but what I used for these estimates is that t-scores are published on the basis of whether they cross the 1.96 threshold. And so what we do is we take the histogram of t-scores and then we remove the effects of that kind of publication bias. Other kinds of publication bias are not accounted for. So that would be a limitation of these. So this simple kind is accounted for.
Yeah, thanks. This is really great. But I'm wondering if you see an extension to thinking about counterfactuals about the other parts of power, the numerator and the denominator, right? So like if you counterfactually had better interventions, you use covariates to reduce the noise, in the standard error, and that got, I'm just thinking about is that sample size is important, right? But there are these other considerations for power, and so you could imagine that might be an extension. So yeah, thanks.
Yeah. So you could actually, mathematically, doubling your sample size or quadrupling your sample size is the same as doubling the effectiveness of every intervention. So you could interpret the results in the exact same way, where actually the estimates are exactly the same if your counterfactual is all of the experiments get square root 2 times more effective.
Thanks so much. This is super interesting. I really like the approach. I feel like I would have chosen a different quantity to simulate at the end, because I feel like part of what you're telling us that this is a measure of how much we've learned from these experiments, but it's really kind of how many discoveries have we made? And I think I'm more interested in a different kind of learning, which is how much have we shrunk, from our priors in a given literature. And so I guess I would have switched to something that's like the mean length of the confidence interval or something like that. And I think the findings might be somewhat different, because what you're finding in part, I think, with the RCTs is not just that the reason why it's not that responsive is a function both of the effect size, the true effect sizes, and the sample size. And so you're kind of saying, for example, in the comparison between RCTs and observational, maybe it's just that we're studying with RCTs a set of things that turn out to not be that effective. And so I want to know something that's I want to know a quantity that's independent of the true effect sizes that would kind of, I feel like that's closer to the value of science that we're, even for null results, we're reducing our uncertainty in how effective things are.
Yeah,
I think like that would be a natural extension of what you have.
Of course. And like one basic fact that you might use is that if we were to multiply our sample sizes by 2, the width of our confidence intervals for most kinds of tests is going to be reduced by 1 over square root of 2, right? But then there's other things like you're really talking about welfare, right? What is our social welfare function here? And how does true effect and sample size map into that? So that's a much bigger question. And for many reasons, and we can talk offline about this, it's a very difficult question.
Oh, very great work. And then a related question, I think you mentioned that there are other papers who developed similar methodologies. How does the result compare to theirs?
So they're also looking at different estimates. So as far as I know, I'm the only one who goes directly after this average statistical power under a counterfactual, right? So I don't have any point estimates to compare to. But what I was saying is there are other studies that study median statistical power, like Ioannidis et al., or others that study that for whom statistical power is an object of interest. And so this is the conversation that I was contributing to. But as far as I know, there isn't a conversation about responsiveness of average statistical power to sample size choice.